The last step in the pre-processing is the summarization. In the summarization, we will try to summarize the peptide intensities into a protein expression value. And then these protein expression values will be used in the next part for differential abundance analysis. We will again use the CPTAC study to motivate the reason for summarization and how we have to summarize. We will use data from lab 2, one lab, condition A and condition E, so the most extreme conditions to really indicate the problems that can arise. And we will do that for a spike-in protein that is differentially abundant across these samples. If we look to the data, we basically see the intensities in the y-axis, and we see the different peptides that have been identified for this protein in the x-axis. And we will first focus on the spike-in concentrations E, the high spike-in conditions, and we clearly see that there are very strong peptide characteristics, so very strong differences in the peptide intensities between the peptides. And this is because these peptides fly differently through the mass spec. We also see that there are some outliers, an outlier here and an outlier there, that we have to take into account. So these do something else than the remaining peptides. If we focus on condition A, the low spike in concentration, we see that only a very few peptides are identified and or also quantified and available. And the reason for that is because the concentration is so low, and we can only pick up peptides that fly very well through the mass spec. So all the peptides that we find have typically high intensities. And all these peptides that have lower intensities in concentration E are not found back for A because they simply cannot be detected. In summarization, we will now try to summarize the information of all these peptides into one protein expression value. And we can, for example, do that by taking the mean. So by taking the mean over all the peptides. And then we have one protein expression value for this particular sample. In our case, we would have three means for the samples E, and we would have three means for one mean for each of the samples A after the summarization. You can already imagine that if you use traditional mean or median summarization, that can introduce problems in the data analysis. We clearly see that the peptides that are not identified in A have different characteristics, and that can introduce biases. In order to accommodate for that, we can come up with more complex summarization methods. We, for instance, could use a linear model where we will try to estimate a sample mean while correcting for the different peptide characteristics. And if we do that, we will correct for the fact that we only find the high peptides in A and that we do not find peptides that fly difficult through the mass spec in this low spike-in concentration. How can we do that? We can do that by a model. We can do that by modeling the log2 transformed intensities for sample I and peptide P with a kind of a sample-specific effect, the summary that we will use at the protein level. And we will now estimate that summary while correcting for the fact that we have peptides from different peptide species. So we will also estimate the peptide effect. And then we will have a sample average that is corrected for peptide species. Before we get into more details, we will first explore these different summarization strategies ourselves on this spike-in study, so that we clearly know what is going on and what the different summarization methods do on real data.